Okay, the pyruvate metabolism we have discussed already, the alanine, the oxaloacetate, the acyl coenzyme, and the lactate. Okay, now we're going to discuss this TCA cycle. Remember, I told you about this acyl coenzyme that it carries a 2 carbon molecules with it, and one is lost in this CO2 and one is lost in this CO2. So there is no net gain or loss of carbon molecules happen whenever a TCA cycle runs. Okay.
Okay.
Now, you should not remember every step of this pathway, but you should, who is this?
Mute the mic.
Mute the mic, please. Good now. So you don't need to remember every step of the cycle, but you just need to remember that this isosaccharic dehydrogenase is actually the rate limiting step. Okay. And this alpha ketoglutarate dehydrogenase is the enzyme that requires the same cofactors as that of this pyruvate dehydrogenase complex. Only this dehydrogenase and this dehydrogenase requires the vitamin B1. This step do not require the vitamin B1. Remember, I told you in the vitamin section, I mentioned about these two enzymes, but I do not mention about this associated dehydrogenase enzyme because it do not require the vitamin B1, but this and this requires a vitamin B1. Please remember this fact. But this associated dehydrogenase complex is actually the rate limiting complex. Okay. Then you should know that the conversion of this succinyl coenzyme A into the succinate forms this GTP molecule. And this GTP molecule can be used as the ATP. And it can also be used by this PEPCK enzyme, which converts this oxaloacetate into the phosphoenol pyruvate, like here. You know, the conversion of this oxaloacetate into the phosphoenol pyruvate is done by the PEPCK enzyme, which is phosphoenol pyruvate carboxylase. And it requires this GTP molecule, which comes from this step, the conversion of succinyl coenzyme A into the succinate. Okay, please remember, they will ask you where this GTP molecule came from, and you should remember this step that produces the GTP. And then you should know about the succinate dehydrogenase complex, which is a complex 2 in your electron transport system. Okay, and this complex requires a vitamin B2, which is your riboflavin. Okay, and then you remember this step about this oxaloacetate. Remember about in the ethanol metabolism that this oxaloacetate will be converted into the malate because you require this NAD stores. You're getting too much NADH, right? And then you should remember about this alanine and the aspartate aminotransferase enzyme, the ALT and the AST enzyme. Both of these enzymes require the vitamin B6 as their cofactor. Okay. And you should know that this alanine will be converted into the pyruvate by the action of enzyme ALT. And this aspartate will be converted into the oxaloacetate by the action of this AST. Please remember, you can write it down in your first aid. Noted? Okay, so please, you can note this point also that this acyl coenzyme enters a TCA cycle and donates its 2 carbon, but those 2 carbons will be eliminated in the form of CO2. And what you will left is no net gain or loss of carbon molecules. Okay, you should remember the fact that this 1, 2, and this 3 NADH formation occur during the TCA cycles. 1 FADH formation occur in this step in the TCA cycle. And 1 GTP molecule is formed during this step. And this GTP molecule is utilized later into the gluconeogenesis. You should remember about this alpha ketoglutarate thing and this isosaccharic dehydrogenase thing. Okay. Any doubt?
Is the ALT AST conversion happening at the same time as the TCA cycle? Because I know the alanine.
No. Whenever you require the aspartate, remember the aspartate shuttle?
Yeah.
This is it.
Okay, you're just putting where it ties in. Thank you.
Yeah, exactly. And this alanine thing will happen whenever you require this alanine amino acid. You will see that later into the later section of this metabolism. about the function of these enzymes also. In the energy of muscles, you will see that alanine aminotransferase function. And these are the two enzymes which are, the ALT and the AST enzyme, they found in the liver, the marker of liver destruction, ALT and AST.
Okay, thank you.
Thank you so much. So we move forward now. No doubt. Okay, then you should remember about this electron transport system. Okay, they have these 5 complexes which named at complex 1, complex 2, complex 3, complex 4, and complex 5. Okay. And you should know that all of the NADH or NAD or FADH, they donate the electron actually. Okay. And all of these complexes are located in the inner mitochondrial membrane. There are 2 membranes are present in the mitochondria, the outer membrane and then the inner membrane. As I can draw a diagram if you want. This is the outer membrane of mitochondria. This is the inner membrane of mitochondria. And this is the inner membrane, this is a space actually, intermembranous space. And after this inner membrane, there is this all the cytosol of the mitochondria. This is your real mitochondria. And all of these complexes located here in this membrane part. Okay. So what will happen as the electron transfers from this complex to this last complex, they will take one hydrogen from this cytosol and will transfer it into the inner membrane step. It's like a feed they are taking. Okay. So NADH here, it will deliver their electrons to the complex 1. From the complex 1, they travel to this complex 3, then complex 4, and then complex 5. And in this process, it will utilizes one hydrogen molecule. It will transfer it from the cytosol or the matrix to the inner membranous space. Okay, you should know the inhibitor of this complex, which is rotenone. Rotenone had this 1 in its name. So you can remember the name easily, rotenone 1. It inhibits the complex 1. Okay. Then comes the second complex, which is a succinate dehydrogenase, the same complex which takes part in the TCA cycle. It uses this FAD, which is a vitamin B2. Okay. It delivers the electron to this cytochrome Q. Cytochrome Q will deliver the electron to this complex 3. And finally, the complex 3 will deliver the electrons to the cytochrome C. Okay. And this is a complex 3. The inhibitor of complex 3 is the anti-mysin. You can remember it as a N3-mysin. Okay, N3-mysin, easy to remember. The anti-mysin A is an inhibitor of this complex. Then comes the complex 4. And this is the complex where the formation of your water molecule and the utilization of the oxygen takes place. The oxidative phosphorylation takes place here in the complex 4. And this complex is inhibited by this cyanide and the CO2. Okay. The cyanide and the CO2 inhibits this complex. Okay. And finally, the complex 5, which is the ATPase complex that takes up the ATP, ADP, and forms the ATP. That's how from the NADH, you're getting the production of this ATP molecule. And the oligomycin is an inhibitor of this complex, complex 5. And you should remember about 2 drugs, which are the 2,4-dinitrophenol and the aspirin overdose. What they will do, they will make this membrane very porous, so that hydrogen can be easily leaked out. So now you require more NAD, you require more energy. Sorry, you require more NAD, you require more oxygen to generate the same level of hydrogen because they are getting leaked out. So what you are getting the waste of energy. And that's why in the aspirin overdose, you get a very high temperature. Remember, we will study that later in parts in the musculoskeletal chapter, that in the aspirin overdose, you will get the fever, very high temperature of your body because of this loss of hydrogen molecules. So you're getting loss of your energy in the form of heat. Okay, any doubt?
Can you repeat the last part along that aspirin overdose?
So when you give a patient aspirin, too much of aspirin overdose, or you give the patient 2,4-dinitrophenol, what these 2 drugs will do, they will make this membrane, the inner mitochondrial membrane, very porous, so that this hydrogen can be easily leaked out. And as this hydrogen leaked out, they will release the energy, and this energy is lost in the form of heat. Okay, that is the first thing. And because this energy is losing in the form of heat, you are getting this high temperature of your body. Okay, and now because you require now more oxygen, you require more NAD, you require more FAD and require more oxygen to generate the same concentration of hydrogen ions now. So in case of the aspirin overdose, you require more oxygen to function. Okay, your breathing will increase. Your breathing will increase. Okay, the CO2 production inside your body will increase. The utilization of the oxygen will increase in the aspirin overdose.
Thank you.
Okay.
Yeah, I can
And
understand.
you should remember this fact that this one NADH molecule will form 2.5 ATP, and this one FADH2 molecule will form 1.5 ATP. Okay. The electron transport inhibitors, I already told you about this rotenone, the complex 1 inhibitor, N3-mycin, the complex 3 inhibitor, the cyanide and the carbon monoxides, the complex 4 inhibitors, okay, and the oligomycin, the complex 5 inhibitor. Now, you guys can see here, if you inhibit the complex 1 by the rotenone, can this ETS will still continue or will it inhibit completely?
Inhibit.
No, it won't inhibit because you still had this FAD, remember, remaining, right? So this ETS will still be continued because you have this FAD remaining. Okay, but once you inhibit this complex 5, you will completely inhibit your ETS system. Okay, remember this fact again. Okay, so giving these drugs will just inhibit or reduces the proton gradient, will reduces the ATP synthesis, but this ETS will still be continued. But if you inhibit this complex 5 using the oligomycin, you will completely inhibit your ATP. There won't be any ATP production occur now because they just lost of everything. Okay. Now the uncoupling agents, I told you about this, the aspirin and 2,4-dinitrophenol, they makes the membrane very porous. They increase the permeability of the membrane so that your hydrogen can be easily leaked out into the matrix again. So you require more oxygen to generate the same hydrogen thing. Okay. You require more oxygen, you will produce more CO2, but your ATP synthesis will be decreased because your hydrogens are being leaked out continuously, right? And they will produce the heat. So you will get this hyperthermia in case of aspirin overdose. Okay. So in case of aspirin overdose, your aerobic respiration will be continued, but it will form less ATP than what should be. And you should also remember about these drugs, the cyanide and the CO2, they actually inhibit the transfer of electron from cytochrome C to cytochrome AA3. This is the complex 4, which is cytochrome CAA3 complex, its name, cytochrome CAA3 complex, and this cyanide or this carbon monoxide. And this is not CO2, this is carbon monoxide. Please change it, guys. It is wrong in FA. This is carbon monoxide, not the carbon dioxide. Okay. So the cyanide and the carbon monoxide will inhibit the transfer of electron from cytochrome C to AA3. Sorry, from AA3 to the oxygen. Remember, this oxygen required the electron. I'm sorry, this is not the cytochrome C, but cytochrome AA3 to the oxygen. They inhibits it. Please remember this fact. NBME is going to ask you this. What is the function of this carbon monoxide and the cyanide? They inhibit the transfer of electron from AA3 complex to the oxygen. And this complex 4 is known as the cytochrome C AA3 complex. Any doubt? Any doubt in this part? Any response would be appreciated, guys.
No, sir, we can move ahead.
Okay, so we will take a short break here for we can expect a short break, right? We can take a break.
Okay. Should we take a break now?
Yes.
Okay.
Okay.
Thank you. We will take a short break of 10 to 15 minutes. Then we will meet again at 6:15. Okay.
Okay.
Okay. Thank you so much, guys.